Hi, my name is Shrina Shah, and I am an M1 student at the NCSM Durham campus. Over the past few weeks, I've had the opportunity to do research at the Duke Human Vaccine Institute studying B-cell responses to the beta-hairpin epitope. Every year, globally recognized organizations such as the World Health Organization attempt to predict which strain of influenza will spread around the world. Manufacturers will then take the selected strain and weaken it to create a vaccine that produces antibodies designed to fight that specific strain. However, if this prediction is incorrect, the vaccine will essentially be rendered ineffective, which puts thousands at risk for infection. Researchers have also found that antibodies have the ability to bind to hemagglutinin, a glycoprotein located on the surface of influenza. Hemagglutinin is comprised of globular head and stem domains. Most antibodies tend to target the head, but these antibodies are often specific in fighting one strain of influenza since the structure of the hemagglutinin head varies significantly with each strain. However, antibodies that can fight against and target the stem of hemagglutinin are able to protect against multiple strains of influenza, but they're very rare. The purpose of this project was to explore how B cells, which produce antibodies, respond to specially designed antigens that only express one part of the hemagglutinin stem called the beta-hairpin epitope. By isolating and studying this one region, we're ensuring that B cells are targeting a region that's similar across multiple strains of influenza. Before this experiment began, 4 mice were immunized with designer antigens, 2 with the antigen S1b22 and 2 with the antigen S1b24. After the mice were immunized, I was given single-cell culture plates where B cells secrete antibodies into a liquid known as a supernatant, which contains organic material that B cells need to survive. After these B cells are cultured, we harvest the supernatant, which may or may not contain immunoglobulin, or antibodies. Then we conduct an ELISA assay that verifies if there is immunoglobulin within a respective well. Wells that test positive for antibodies are analyzed using a Luminex assay, where we measure mean fluorescence intensity, or MFI, to determine how strongly each antibody binds to a bead coated with a different antigen. Essentially, this helps us identify cross-reactive beta-hairpin specific antibodies. To help interpret our results, we used a control plate with antibodies that weren't expected to bind to either the head or the stem of these antigens. For each antigen, we then calculated the average MFI value and multiplied it by 10 to establish a threshold for meaningful binding. Any antibody above this threshold was considered bound to the antigen. Among the 2 mice immunized with S1b22, we identified a total of 809 immunoglobulin positive cultures. In mouse 1, 61.3% of the cultures showed significant binding to the antigen, and in mouse 2, 43.9% of its thresholds met this threshold. For the 2 mice immunized with S1b24, there were a total of 657 immunoglobulin positive cultures. In mouse 3, 33.9% of its cultures demonstrated significant binding, and for mouse 4, 72.1% of its cultures demonstrated significant binding. While a majority of these antibodies were only able to bind to the antigens the mice were immunized with, we did observe cross-reactivity. In mice 1 and 2, which were again immunized with S1b22, there were 33 cultures with sufficient binding to S1b24, and in mice 3 and 4, which were immunized with S1b24, 7 cultures were able to bind to S1b22. Additionally, when we analyzed the data across the other antigens tested in the Luminex assay, there were 7 cultures with sufficient binding to the antigen H3X3168EHA2, 3 cultures with sufficient binding to B Malaysia Q4EHA2, and 6 cultures with sufficient binding to the antigen H3HK68 full, which suggests that some antibodies are able to bind to the beta-hairpin epitope and not just the scaffold, which holds the epitope. In conclusion, our findings show that these designer antigens are able to stimulate B cells that produce antibodies, some of which are potentially cross-reactive. Our research suggests that these immunogens are not only prompting strong responses to the scaffold, but it may also be possible to redirect B cells towards creating antibodies that bind with the beta-hairpin epitope, which puts us one step closer towards developing a universal influenza vaccine. I'd like to take a moment to thank the NCSSM Foundation, the Burroughs Welcome Fund, the SHRIP staff, and my mentors for giving me such a wonderful opportunity. Thank you for watching.